Hello and welcome to G Fun Facts Online, the show that dives deep into the amazing science all around us. Today we're talking about war, but not the kind you're thinking of. This is a secret war, waged silently for decades, not with bombs or bullets, but with birth control. We're going to uncover one of the most elegant and ingenious strategies ever deployed in the fight against pests that threaten our food and our health. It's a method that turns an enemy's greatest strength against itself. This is the incredible story of the Sterile Insect Technique, or SIT. So to understand this brilliant idea, we have to go back to the 1930s and meet our villain, the New World screwworm. And trust me, this bug is straight out of a horror movie. It's a fly that lays its eggs in the open wounds of animals, cows, sheep, even people. The larvae then hatch and, well, they eat the living flesh of their host. It was a gruesome, painful end for the animal, and it was costing the American livestock industry millions upon millions of dollars every year. The situation was desperate. Enter our heroes, two American entomologists, Edward Knipling and Raymond Bushland. They were studying the screwworm and noticed two very important things. First, the male flies were extremely, let's say, aggressive in their pursuit of a mate. And second, the female screwworm typically only mates once in her entire life. This sparked a revolutionary idea. Knipling thought, what if we could sterilize the males? What if we could raise millions of them, make them unable to have kids, and then just flood the environment with them? These sterile males, being just as aggressive as their wild cousins, would go out and compete for the females. A female who mates with one of our sterile males would lay her eggs, but nothing would ever hatch. She'd have done her one-and-done mating, and her reproductive line would end right there. It was a concept of autocidal control, using the insect to cause its own destruction. The only problem was, how do you sterilize a fly without killing it or making it unattractive? The answer came after World War II, with the dawn of the atomic age: radiation. Scientists discovered that a careful dose of gamma rays or X-rays could damage the reproductive cells of the insects, making them sterile, but leaving them otherwise healthy, strong, and ready to mingle. With the theory and the technology in place, it was time for the ultimate test. In the 1950s, after a small successful trial on an island in Florida, the program went big. They launched a full-scale eradication effort in the southeastern United States. They built massive facilities, essentially fly factories, that produced billions of sterile male screwworms. These sterile flies were then systematically released from airplanes over the affected areas, and the result? It was a spectacular success. By 1959, the New World screwworm was completely gone from the southeastern states. The livestock industry was saved an estimated $20 million a year, which is a huge amount for that time. The program was so successful, it was expanded to the southwestern US, then Mexico, then all the way down through Central America. By 1991, Mexico was declared screwworm-free, saving their industry an estimated $2 billion. Today, there's a permanent barrier of sterile flies being released in Panama to keep the North free of this horrible pest. It remains one of the greatest success stories in the history of pest management. So how exactly do you pull off something this massive? Let's break down the Sterile Insect Technique. Think of it as a 5-step high-tech operation. Step 1. Mass rearing. You need a factory, a bug factory. You have to create the perfect conditions to raise literally billions of the target insect. This means developing artificial food, controlling the temperature and humidity, and basically tricking these bugs into thinking they're in paradise so they can multiply like crazy. Step 2. Sex sorting. In many cases, you only want to release the males. For pests like mosquitoes, it's the females that bite and transmit diseases. So releasing females would be counterproductive. Scientists have developed clever ways to sort the boys from the girls, sometimes using genetic tricks to make it easier. Step 3. Sterilization. Once the insects are at the right stage, usually as pupae, they get a quick, carefully measured dose of radiation. This is the key part. It doesn't make them radioactive. It just damages the chromosomes in their sperm. So when they mate, the sperm can still fertilize an egg, but the damaged genetic code means the embryo can't develop. No babies. Step 4. Quality control. This is crucial. You can't just release a bunch of weak, clumsy flies. They have to be top-tier specimens, able to fly well, live long enough, and out-compete the wild males. So they go through rigorous testing to make sure they're fit for the mission. Step 5. Release and monitor. The sterile insects are packaged up, often chilled to make them inactive, and dropped from planes using GPS to ensure even coverage over the target zone. After release, scientists set traps to monitor the population, checking the ratio of sterile males to wild males and making sure the number of viable eggs in the wild is dropping. It's a finely tuned, data-driven process from start to finish. And the screwworm isn't the only success story. This technique has been a game changer all over the world. Take the Mediterranean fruit fly, or medfly. This pest loves over 250 types of fruits and vegetables. An infestation in California could wreck their multi-billion-dollar agricultural industry. For decades, California has run a medfly SIT program, releasing hundreds of millions of sterile flies every week as a preventative shield. It has reduced potential outbreaks by over 98%. In Africa, the Tsetse fly transmits a devastating disease called sleeping sickness to humans, and a similar disease to cattle, making huge areas of fertile land unusable. SIT was a key part of a program that completely eradicated the Tsetse fly from the island of Zanzibar, a landmark victory for public and animal health. In Canada, apple and pear farmers used to battle the codling moth with tons of pesticides. Now, an SIT program in British Columbia has cut insecticide use against this pest by over 95%. The benefits are huge. Environmentally, it's incredibly precise. It only targets the pest species, leaving beneficial insects like bees and butterflies totally unharmed. And because you're not spraying chemicals everywhere, there are no nasty residues on our food. Economically, it means higher crop yields, healthier livestock, and access to international markets that have strict rules about pests and pesticides. Now, as amazing as SIT is, it's not a magic wand for every pest problem. It's a high-tech approach with some serious hurdles. First, it's expensive to set up. Building a massive bug factory and funding the logistics of a huge release program takes a big initial investment. It's also incredibly complex. You need highly trained people and a smooth operation to rear and release billions of healthy, competitive insects week after week. And there's even a small risk that the wild insects could evolve to resist the technique. Some wild females might develop a preference for wild males over the lab-reared ones. But science doesn't stand still. The future of SIT is even more exciting and looks like something straight out of science fiction. Instead of radiation, scientists are now using genetic engineering. One technique is called RIDL, which stands for Release of Insects with a Dominant Lethal Gene. Basically, they create male insects with a self-destruct gene that gets passed to their offspring. The offspring can't survive without a special antidote that only exists in the lab, so no radiation damage, which can make the males even more competitive. Then there's the even more advanced and controversial idea of a gene drive. This is a genetic tool that can rapidly spread a trait, like sterility, through an entire population, potentially wiping it out. It's incredibly powerful, but also comes with big ethical questions. On top of that, automation and AI are making the whole process cheaper and more efficient, from robotic bug rearing to AI-powered release strategies. The sterile insect technique is a powerful testament to human ingenuity. It's a story of how deeply understanding the biology of another creature allowed us to develop a solution that is targeted, sustainable, and far gentler on the planet than the chemical alternatives. It's a war waged not with poison, but with knowledge. As we look to a future with a growing population, a changing climate, and constant threats to our food supply, Innovations like SIT will be more critical than ever. It shows us that sometimes the most effective way to solve a big problem is with a very, very smart and very tiny solution. Thanks for tuning in to G Fun Facts Online. Join us next time for another dive into the science that shapes our world.